Hello everyone. Today we are going to learn how to count cells in a hemocytometer. So this is the hemocytometer. To count cells on the hemocytometer, there are some basic requirements that we need. These are a culture, um, cell culture or a bacterial culture, a Trypan blue stain, phosphate buffer saline, and a hemocytometer. So for the viability check of the cell, so first we need to prepare the Trypan blue to 0.4% in 1x pps. So we need to prepare the Trypan blue first and maintain the pH at 7.2 to 7.3. Now this 0.1 ml of Trypan blue from this is mixed with 0.1 ml of cell culture and it is gently mixed again with the pipette. From this 10 to 15 microliter of it is given on this hemocytometer and is examined immediately at 10x. Okay. So what does this Trypan blue do actually? Why are we using this Trypan blue? So this Trypan blue is basically a stain that is frequently employed to differentiate between the viable and the non-viable cells. So we can say that it distinguishes between the dead cells and the live cells if we simplify it. Okay. So Trypan blue is widely used for this purpose. Now it is a type of acid azo dye and it belongs to the synthetic dye family and they are not found in nature obviously. So why they are called azodyes? Because they are the organic compounds which have the first functional group like this, that is R bond N double bond N bond R dash. Okay, where this R and R dash are aryl and substituted aryl groups respectively, so that is why they are called azodyes. And since they have this bond, they are very economic and they are very widely employed for this viability check purpose. Okay. Now this Trypan blue, what it does, it stains the dead cell in blue color. It will appear like blue. Whereas the live cells will appear clear. So this whole Trypan blue staining depends on the membrane integrity of the cells. Okay. So the cell membrane of the live cells will remain intact. Therefore, the Trypan blue cannot penetrate inside the cells and therefore it will appear clear. So this is the principle of the Trypan blue. Okay. Since the membrane is intact, the Trypan blue will not get inside of the cell. That's why they will appear clear. Whereas in the dead cell, the cell membrane will get distorted. That is why the Trypan blue will penetrate inside the cells and will give it a blue color. Okay. So if we simplify it, then we can say that the dead cells will appear blue in color, whereas the live cells will appear colorless. Okay. So now, how these Newbus chambers look like? So if we focus on the Newbus chamber, we can see there are two places, the same chambers on both sides. You can use any one of them. And if we enlarge one of it, we can see that this chamber looks somewhat like this. Okay. So here we can see there are total 9 boxes in this chamber. And there are 4 large boxes at the corner of this chamber, okay, which are colored in blue color. And inside these 4 large boxes, you can see there are 16 squares inside each of the boxes. Okay, so there are 16 squares. However, in the middle, we can see that there is a box in the middle of the 9 squares which has 25 squares inside it. Okay, you can count it. There are 25 squares inside it. Now, each of these large big squares, they have a surface area of 1 mm square and a depth of 0.1 mm. Okay, so why this Newbus chamber is made like this means different types of chambers, 16 squares. Some is divided into 16 squares, some is divided into 25. Why this kind of chamber is made? So the reason behind this is that we can count the different types of cells in this chamber. Okay, so microbial cells, okay, there are mammalian cells. So whatever type of cells it may be, it can be counted in the same chamber. Okay. So eukaryotic cells, if you see, they are counted in this big squares which are colored in blue colored. Okay, which have the 16 small squares inside it. So they are counted in this blue squares. Since they are macroscopic, they will count in the blue squares. Okay, which have the area of 1 mm square and depth 0.1 mm. Whereas the microbial cultures, they are counted in the middle of the 9 big squares, which is colored in green color, which contains 25 small squares. So microbial cells are 10 times smaller than that of the eukaryotic cells, so we cannot count it in the larger ones. We need the smaller ones. So because of it, the middle green color one we'll use for the microbial counting. Okay. So the area here is 0.04 mm square and the depth is the same 0.1 mm. So now what we see, there are some rules and regulations that we follow while counting the cells in this hemocytometer. Okay. So here you can clearly see that the colorless cells are the live cells and the stained cells are the dead cells. However, there are some cells that are present on the borderline of the boxes. Okay. So this borderline, the cells that are present on the borderline, they are not counted. So here, do not count the cells on the borderline of the right side of the square in the bottom that are present on the right side and the bottom of the boxes. They are not taken into account. Okay. In all the boxes, you do the same. So these are discarded. Why? In order to avoid the overlapping of the counting of the cells. Okay, so that is why it is avoided. So you need to count just the cells that are present exactly inside the boxes, not in the borderline. Okay, particularly in the right side and the bottom side, we need to avoid this one. Okay, so there are some formulas that are given for this counting. That is, if you want to find the total number of the viable cells, we need to count the colorless cells or the non-blue cells in 4 of the corner squares. Okay, and in order to find the total number of non-viable cells, we need to count the colored cells in the 4 corners. So by adding these two, we will get the total number of cells. So the total number of cells equal to total number of viable cells plus total number of non-viable cells. Okay. Now, if you want to find the average number of the viable or non-viable cells per square, we need to divide the total number of the viable or non-viable cells divided by the total number of squares counted. Okay, suppose we are counting on the 4 large squares, then we have to divide it by 4, for example. Okay, this will become more clearer when we see an example. First, we cover up the formula here. Second is, if you want to find the percentage of the viable or non-viable cells, then we need to divide the total number of viable or non-viable cells by total number of cells, that is both live and dead, into 100. This will give the percentage of viable or non-viable cells. Now coming to the cell density, that is how many cells are there per ml. So for this, we multiply the average number of viable cells by into dilution factor and divided by the volume of a square. So now volume of a square in the 4 large squares, remember the surface area was how much? 1 millimeter square and the depth was 0.1. So the volume becomes surface area into depth. Okay. So this is the cell density. And this cell density is again simplified as average number of viable cells into dilution factor into 10 to the power 4. So if we find out the volume of the square, it will somehow become 10 to the power 4. So this is the simplified form. Okay. Now coming to the dilution factor. So the dilution factor, how to find the dilution factor is that divide the final volume by the sample volume which we have taken. And the total number of live cells, if you want to find out, suppose in certain amount of sample, we need to find the total number of live cells. So what we do, we multiply the cell density which we got, cells per ml, cell density, into the original volume of sample which is taken in ml. Okay, we just multiply it and then we can get the total cell number present in our sample. So coming to this, how to read in hemocytometer, actually, how to count the cells. Okay, so for counting the cells in the large blue boxes, we have 2 types of patterns. Okay, so the first is going from left to right, then downward and again right to left. So this is one type of pattern. And the second pattern is going left to right, then down a little bit, then again right to left, then down a little bit, then again left to right. So this is in a zigzag motion you can read. Okay, so in order to remember which boxes you have finished counting. Okay, so in order to avoid this confusion, if you are counting the cells in the middle green box, you just do the same thing, read it in a zigzag manner so that you can remember which boxes you have already counted. Okay, so this is the most preferable pattern that is used for counting the cells in the hemocytometer. Okay, now let us come to the large blue boxes. Okay, let us count some cells in the large blue boxes with an example. So suppose this is the hemocytometer and it has the cells in the blue boxes. Okay, so we need to count it. So let's say there are this much amount of viable and dead cell present in the four corners. That is this large blue boxes. So this much amount of viable and dead cells are present. Okay, so we can easily find out the total number of viable cells. That is adding all this up, 23, 18, 22 and 29, which gives 92. And the total number of non-viable cells becomes 3 plus 4 plus 1 plus 4 equals to 12. Okay, it's very easy to find out. Okay, now coming to the average number of viable or non-viable cells. Okay, so we'll find out the average number of viable cells for the same these things only. So the average number of viable cells per square becomes 92 divided by 4. Remember the number of the squares you have counted, that is 4, right? So it becomes 23. Now if you want to find out the percentage of the viable cells per square, we need the total number of viable cells divided by total cells into 100. Okay, so viable cells was 92 divided by total cells, that is viable plus non-viable, that is 104 into 100, which gives 89%. So we can say there are 89% of viable cell present per square, right? Now if we find out the cell density or the concentration of the cells, that is average number of viable cells into dilution factor into 10 to the power 4, right? Just we have studied in the previous slide only. So it gives 4.6 into 10 to the power 5 cells per ml. So this is the cell density. Now if suppose we have 5 ml of sample, so how to find the total number of live cells in the 5 ml of sample? So for this we need to multiply the cell density with the original number of sample, original volume of sample, sorry. So that gives 4.6 into 10 to the power 5 into 5. Suppose if we have 5 ml of sample, right? So it becomes 2.3 into 10 to the power 6 cells. So we can say there are this much amount of cells present in our 5 ml of sample. Okay, clear? So this is how we calculate. Now this volume of square here, you have to note that volume of square is surface area into depth, which is 1 mm square into 0.1 mm. Okay. Now coming to the middle green boxes. Suppose we have to count the cells in the middle green box. This one. Okay. This is the enlarged view. Suppose we have this much of viable and dead cells available in this green box. Okay, microbial cells. So we'll count these corner boxes and the middle one in a zigzag manner, right? We have seen in our previous slide. So we will count the corner boxes and the middle boxes. So it's very easy to find the total number of the viable cells. Okay, just add all this up. So together it gives 115 and total number of non-viable cells here becomes 50. Okay, now coming to the average number of viable cells in the same box, if you find, that is average number of viable cells per square becomes total number of viable cell divided by the number of boxes counted. So 115 divided by 5 boxes counted, so divided by 5. So it gives 23. Now if we find out the percentage of the viable cells per square, that is total number of viable cells divided by total number of cells into 100, which gives 115 divided by 130 into 100, that gives 89%. So here also we can say that there is 89% of cells present per square, right? Now coming to the concentration of the viable cells per ml, or we can just say cell density. So this formula is average number of viable cells into dilution factor divided by the volume of a square, right? So here the average number of viable cells is 23, dilution factor is 2, and the volume of square is 4 into 10 to the power minus 6. How? Here we see there is a note, volume of square is 0.04 mm square into 0.1. Okay, so if we calculate this and we convert this into centimeter and then into ml, so all this calculation will give 4 into 10 to the power minus 6. Okay, so if we calculate this one, we will get 11.5 into 10 to the power 6 cells per ml. So the cell density we can say 11.5 into 10 to the power 6 cells per ml, or 1.15 into 10 to the power 7 cells per ml. We can also write like that. Okay. So suppose we have a total of 5 ml sample and we need to find the total number of live cells in the 5 ml sample. Okay. So what do? We will simply multiply the cell density with the original volume of sample, that is 11.5 into 10 to the power 6 into 5, which gives 5.75 into 10 to the power 7 cells. So we can say that there are 5.75 into 10 to the power 7 cells in a 5 ml of sample which we have taken. Right. So this is how the counting of cells in the hemocytometer is done. I hope you have your concept clear. You have got your concept clear by this. So here we have learned about the counting of this hemocytometer, counting cells in this hemocytometer, both in the large boxes and the middle boxes.